Thank you, Chintong. It's very kind of you. I wouldn't say I'm an expert lah. Let's say I spend a little bit more time and read on all that. Okay, this research also I think is very good. It give you a neat paradigm. Okay, it's featured in Ganoderma feature 76, issue 76. Okay, the heading of the title is like it's Network Pharmacology Analysis. Okay, of the Ganoderma Lucidum extract show that it has multiple anti-liver cancer targets. Okay, can we move to the next slide? Okay, there will be the heading network pharmacology. When I come across network pharmacology, I think, what network? You know, network is like, is it like our networking MLM business? Okay, first point. All right. I mean, we know that cancer is a worldwide health problem. Okay, and it's among the leading causes of death. Probably among second or third causes of death in the world. Everywhere, the statistics have shown that every 3 or 4 person, 1% will suffer with surgical resection, you know, all that. But the next point, it shows that the cancer mortality rate is still for those has a relative 5-year survival rate, still not there. They are still not improved despite all these new therapeutic methods being introduced. You know, I have heard of like in 2010 they talk about immunotherapy, all that. Also seems to have a problem. All right. Okay, so over the last 5 years, I mean, I would say that it's despite all these new therapeutic methods, the mortality rate or survival rate of 5 years and above has not improved. I mean, naturally, normally for cancer, they measure in terms of 5 years. So if the treatment, you go through a treatment, you survive more than 5 years, then it's considered successful to them. Alright? But of course, we also have to realize that there are different stages of it. But this one is from the study done by Alemany in this global surveillance of cancer survival 1995 to 2009. That is statistics. Alright? Okay, next. Okay, just now I mentioned about targeted drugs, photodynamic therapy, intentional therapy. I just want to have a definition in case people who do not know what it is. A targeted drug as it is just basically target a particular gene or protein in your body to try to cure or try to treat the cancer. Photodynamic is basically a special treatment where they inject some photosensitizing agent into your bloodstream, and then as it goes along, they kill it. Then interventional therapy is also something like that. It's a subspecialty. Same thing also, we also do a certain minimal invasive, something like laser or what, go in, and then they see the polyps and they kill it. Then they burn it, or something of that sort. Okay? Right, next one. So, given the fact that they have not been able to successful in discovering cancer drugs that can allow improvement in the 5-year survival rate. Therefore, the researchers are looking cure the disease. This is something like what you see in targeted drugs. Okay, of course, when you target a particular drug, you avoid all the side effects. Compared to like people who go through chemotherapy, there's a lot of side effect, right? Vomiting, ulcer, cannot swallow, all that. These are all side effects. So if they can target a particular drug, the side effect is lesser. Okay, that is the case. But unfortunately, the efficacy and the unexpected toxicity of such drugs, right? Okay, cancer and also like most chronic diseases, uh, like diabetes, metabolic syndrome, or they are all not due to a dysfunction of one particular molecule in the body, one particular genes, you want to say one particular gene, but rather it is caused by the dysfunction of the whole related network, right? So as a result, therefore, when we want to diagnose and treat cancer or also chronic diseases, we should be not looking at one particular gene, but maybe a whole pathway, a whole system of the body. That's why they call it network, whole network of the body. Okay? That's why the term network pharmacology analysis come about, network. Alright? Okay, so therefore the new trend towards drug discovery will be those that are able to act on multiple proteins instead of just particular one area. Alright, one particular gene that we're looking at. So it's like if you I remember when I was when AIDS came into the scene in the 80s, last century, 1980s and above. They were having problems, and then until Thailand come out with a cocktail of drugs to try to cure it. So now, start using such multiple uh, protein drugs to cure a particular disease has come into. Okay? Right, next slide, please. Okay, so we now introduce the new concept, network pharmacology. Alright, this network pharmacology is trying to explore the potential of drugs that can act on multiple proteins, multiple targets. Right? Targets are multiple protein in the body instead of one particular gene. Okay? Now, this network pharmacology is a very wide field. It basically uses computerized method that combines system biology and polypharmacology. When I was a student in the 70s, uh, we talk about RNA and DNA ready in the business magazine. But they cannot continue further from there. Why? Because during that time, the best computer, top line computer was only a 64 kilobyte processor. So when they try to introduce the human genome, they find that they cannot track the whole body biological system. Alright? Although they know that it's all related to one another, but they cannot so-called set up the link completely. Although they know that it's all related, the body system is all related to one another. Okay? Then you have polypharmacology. Like just now I mentioned, in Thailand, where when it comes to AIDS, they find that they are not able to have one particular drug cure to treat the AIDS patient. They need to have a cocktail of drugs. A cocktail of drugs that target different proteins in the body. So that's why you have this polypharmacology. So when these two combine together, plus the the disease target and know the mechanism and pathway and the relation between them. Okay? Right, next slide. Next slide. Alright, so now we come back to our Ganoderma Lucidum. Now that we know what is network pharmacology, network pharmacology, of course, we will first have to identify what we want to study. The first thing we know that Ganoderma, we know that it has been used in Asia for a long time, right? To treat tumors and to treat chronic diseases, right? And we always hear that Ganoderma lucidum has multiple compounds like peptidoglycan, polysaccharides, and all that. Peptidoglycan are a group of carotenic acid, and they have multiple targets against tumor and chronic diseases. But unfortunately, a lot of all these the studies made were all made for individuals. Alright? Now, when the researcher studied Ganoderma in the past, they always pick a particular compound and then they add on the protein. Right? Then, when the protein is effective, they say, oh, it's effective against this particular disease or this particular illness. And then they combine with other research that is also may not be of the same compound, but of different Ganoderma, may be also from different species, and then they say Ganoderma therefore has multi-target. Alright? Okay, so this particular study, next slide please. So this particular study is want to prove that Ganoderma itself from one particular species that's most commonly cultivated, Ganoderma lucidum. They also take into account Ganoderma sinensis, and then to see the extract from this particular species, whether itself has this multiple targets, and at the same time understand the mechanism of its anti-cancer therapeutic action. Okay? This particular study paper titled, next point, is Network Pharmacology Analysis of Anti-Cancer Pharmacological Mechanism of Ganoderma Lucidum Extract with Experimental Support using Kappa 1,6 bearing mice. It was published in the Journal of Ethnopharmacology in 2017. Alright? So the study, the researchers were from Shanghai PCN University. Okay, in China, there were, I think, a few PCN universities there. Alright, okay, now that we have identified the product in the study, of course, eventually, we want to know what are the compounds inside that particular product. The next slide. So, the compound that we can find in this particular Ganoderma lucidum extract, and then if you have visited our factory in Taiwan, Dr. Cheng always talk about this PLC identification. Of course, when you extract a certain compound, you have to confirm the existence of such compound with this HPLC machine to say that this compound exists. So this particular research did it other than HPLC, ELSS, MMR, I don't know what is this, but it's something like they confirm that this is all the compound that exists in Ganoderma lucidum. Okay, they have managed to identify 12 active compounds, right? Ganodermic acid A, B, C2, BH1, and then ganodermic acid ABY, and camphorol, and all the chemical names. Alright, so these 12 active compounds researcher go through the library, and they find that this drug compound has been related to all the anti-cancer properties. Okay, maybe let's go through the next two slides and see. Next slide. So you can see Ganoderma has to do with suppressed cell proliferation in various cancers, including hepatocellular carcinoma. Alright, Ganoderma ACP has the potential to develop into anti-drug resistance. H also suppress proliferation, colony formation, invasive behavior of breast cancer, all that. Okay, and then next one, galactonic acid C2 inhibit DNA fragmentation. So especially when we go for chemotherapy, your body DNA formation is completely fragmented. You completely become weak, and that kind of thing. All right. So you also has hepatoprotective effect. All right, on the liver. Ganoderma ACP also inhibit proliferation, cervical carcinoma. Why? A biological effect on cholesterol synthesis in human hepatitis cells, right? Lower blood cholesterol. Then the other immune hypothesis, and the cell kill the cell. Okay, proliferation inhibition in lymphoma, colon cancer. The genistein inhibit cell renewal ability of the cancer stem cell. Right? So all these, as you can see, were related to some cancer cells. Okay? Next one. So I will not go through more on this, but basically it's all related to cancer activity. So all these studies were already done. So the 12 compounds that they have identified is from Ganoderma Lucidum has already been proven individually in all these studies. Okay? Next one. So after they have identified the compound in the Ganoderma Lucidum, they have to know whether this compound can so-called bind to the protein in our body. It's just like right now COVID-19, we talk about the virus, how it binds to our body, and then it spread from there, right? So same thing is also has to come about. How can this compound bind to the protein or genes in our body? Okay, so they do it. They feed this drug compound into a computer. This computer will run through the library like PubChem, ECMSP, all the drug bank, all these compounds. Okay, they run through and then they find out that they are 20 potential targets. Right, 20 potential protein, you want to say that way, or genes, or messenger RNA, that these compounds from Ganoderma that can bind it to, okay, to act on it. Alright? Then, of course, you don't just simply take all the top compounds and the compounds, because some only bind one or two. Alright? But in order to show a more effective method, we only choose those compounds that can bind to 3 or more of the target. Right? Then as a result, they arrive at 6 targets. 6 targets that bind by these 7 active compounds. Okay, 6 targets. When I say target, I mean genes, explanation. So you can see Ganoderma Lucidum has the compound, and this compound can hit all these targets. Actually, if you look at it that way, it's just another way of explaining network pharmacology. Before network pharmacology, just now I was telling you about the paradigm of the current drug discovery, that of a single drug, single gene, single disease. It's just the same as maybe you use a metaphor that says like using a magic bullet. A magic bullet to kill the disease, to cure the disease. One bullet to cure one. Right? So that thinking is, as you can see, is not so effective. So the thinking should therefore change it from one bullet, instead of one bullet, it changed into a tablet or sharpener. Right? When we look at those, okay. So this one, of course, is also a definition to tell you more about the six targets that we talk about. Alright, that of NRCI, which are what are they? They are associated with childhood acute lymphoblastic leukemia. So they are also related to cancer. Alright, all these six targets, right? Like NR3C2, which is in long form is nuclear receptor subfamily T group C, number 2. They are associated with poor prognosis and survival in pancreatic cancer. Alright? Like progesterone are related to breast cancer, uterine cancer, chorionic receptor are related to non-small cell lung cancer, because blocking this chorionic acid cause the inactivation of the NF-kappaB that reduce the migratory effect of the basic capacity of the tumor cell. Okay? So there were studies done in all these proteins, all these genes. They are all related to cancer. Right? So therefore, we have I would say the computer docking shows that there is a linkage between Ganoderma and be able to bind to this particular gene or this particular messenger RNA that is related to cancer. So there is a linkage. Alright? So like estrogen receptor 1, it is a transcription factor and therapeutic pertaining targeting hormone-dependent female cancer. Alright, androgen receptor involved in tumor progression, such as cell proliferation, all that kind of thing. Okay, so they have done a biological analysis. All in all, maybe just now the use definition slide, then you may see already a little bit more or very confusing, but in general, they are all involving about signaling pathways, anti-cancer signaling pathways. So all these 6 targets, right, they all involve signaling pathways, anti-cancer. They have to do with cancer, okay? Like tumor necrosis signaling pathway. It has to do with apoptosis, right? That is the cell dying off itself, inflammation, immune response, estrogen signaling pathway is related to lung and liver cancer, and also breast cancer, all that kind of thing. H1, F1 signaling pathway, right? All related to the growth and proliferation of tumors. Next one is neutrophil signaling pathway, like maintenance of cell survival, apoptosis. So all this you can see that all these signaling pathways are related to cancer, right? They are one way or the other. If you target all this, somehow or other you can improve the cancer, right? So there is a correlation. We find that okay, they can bind to all this. So as a result, they have concluded that these are the right targets that they have achieved. The computer has done it correctly, right? That means with the compound that they put into the docking program, and the target and or genes or DNA that they match to, that they can bind, confirm that the compound in Ganoderma has anti-cancer effects. Okay, so but of course the researchers will just sit down there and say, oh, we are happy when that is the case. They confirm it through with other programs like blood bank PubChem and they search through all these public database and look for whether the compound that they identify is it correct, the target that they identify is it correct. So through the same computer program that they go through, they find that Ganoderma Lucidum actually has 122 effective compounds. Okay, they have screened through 122 effective compounds, out of which 160 targets were deleted. And as a result, they find that 18 of them are important. And of these 18 of them are pertaining to cancer, the six earlier that they have selected was within the 18. So therefore, the researcher concluded that Ganoderma Lucidum extract has anti-cancer effect and it plays an important role in this anti-cancer treatment. Right? Okay, this is just on paper, on computer, that they do it. Right? So they have to confirm that their findings is correct. Right? Their finding already proved that the compounds in Ganoderma has anti-cancer effect, right? Because we already know that there are multiple targets from all that study that they have made. Okay, so they do animal study. Right? So they do an animal study. Right? So to do an animal study, to confirm that this active compound and target that they have targeted is actually working, is true, not on just paper only. Alright? So therefore they will plant some cells. Like I wrote down here, logarithmic case, and it's the cell is growing. You have to cultivate the cells. Okay? These cells is a Apple 1/6 cell. Apple 1/6 cell is a cell that is closely related to liver cancer. Of course, this is not a cell that is extracted from the liver cancer, but it is a closely related cancer cell that is commonly used to study liver cancer. Okay, it's closely related, right? They then implant these cells into the underarm of the rats. Okay, of the rat. Okay, the underarm. Okay, underarm under the thigh, in the inside of the thigh, the soft part where they can easily grow. Okay, the amount that we inject in all this mice is equivalent to 15 grams of GLP for 60 kilo man. So I was just doing a calculation, based on our 用箭 if you are 60 kilo, you are looking at cancer treatment, you're talking about 33 tablets, equivalent to 33 tablets. So was injected into the mice from day one. The moment they inject the cell, at the same time, they also feed Ganoderma extract to the rat from day one for 28 days. Okay? And 1 week after inoculation, and every 3 days, they will measure the tumor volume, the weight, okay, how it goes. Right? Next one. Okay, so after 28 days, the result. You see how it happened. Alright, they do a pathological analysis of the tumor, and they find out that in the Ganoderma extract group, large area of the tumor was having tumor necrosis. In the tumor of the GLE group, there were large area of tumor necrosis, dead cells, and surrounded by inflammatory cells. Inflammation is generally good. Short-term inflammation is good, because when inflammation, that means something has a cut, you have an infection, have a growth, your body immune system come and attack it, right? So there's a lot of inflammation. For short-term is good, right? Because this inflammation will, like killer cell, neutrophil, macrophages, all this will come and eat up all these cancer cells, right? So, in a sense, this point that we mentioned also proves that when they plant the cell, that means it's correct, it's growing, right? That's why you have all this inflammation in the body, right? Then, at the same time, when we measure the tumor of the GLE group, and compare it with the model group, the group that has no treatment, they find that the tumor size was only 1/3 of the model group, and the tumor weight was half, and this is smaller, and it's not growing as fast. Okay, in cancer, anything that growing at the rate of less than 15%, if I'm not wrong, or 12%, is considered elimination, something like that, So this shows that Ganoderma Lucidum can inhibit tumor inside the body. Alright, in vivo. Okay, I have a graphic representation in the next slide to show you the compare. Okay, you can see that in the top left corner, the picture, the model group where the rats is not fed with Ganoderma, the tumor group is bigger. Where those with fed with Ganoderma Lucidum except the tumor size is smaller. So the growth rate is half of it. The C shows. Okay? The B is just is a histologic image to show the inflammation, which I'm not able to interpret so but it's there. Okay? So, of course, although we say that, correct, grow like that, lesser, but we pick those 6 targets. Are they really that effective? Are they really working through that 6 targets? Therefore, in order to verify that, okay, the researcher do a quantitative reverse transcription and whole chain PCR. Just like right now COVID-19, right? You want to count the number of cells, you take a PCR test. You take a saliva, they put into your mouth, take the saliva, then after that they look for count the PCR, so something like that. So this one we do a reverse one. Okay? So statistically you have to hold constant one group. Okay, so then therefore they unitize the model group. Okay, they convert all the model group to one. And then when the GL group, they based on the same basis and work on the correlation as a relative mathematical model. And if they find that there is a difference, right, when you hold one constant and then you work the other accordingly, this one mathematics, probably those people who are mathematicians will be able to explain clearly. Shows that there is a difference in this factor. So since there is a difference in this factor, this go to prove that Ganoderma Lucidum extract, right, really act to these 6 targets. Alright, that means through these 6 targets, you can show the effect. Okay, so the B one is a bit difficult to understand now because we are laymen, don't know what C show a slight difference, right? Of some, right? Okay, okay. Now we come to the conclusion, right? So the conclusion shows that the active compounds found in Ganoderma can act to multiple targets to treat cancer. Okay, and network pharmacology analysis allow us to understand the pharmacological mechanism of GLE. It gives us that scientific explanation. Or else we always say that this pantotenoid polysaccharide can act like that. How it act like that? So network pharmacology tells us that it can act through these 6 targets that you mentioned. Alright? It's just like I mentioned just now, like a single bullet. You take a single bullet, you try to shoot one, you only shoot one particular gene. Is it that effective? As compared to you shoot one, a pellet appear, a bird appear, a bird, few birds on the tree, one shot you can kill all. Something like that. Okay? So therefore, if multiple target drugs are seen as an evolutionary product, like just now I mentioned, it's an evolutionary thing because right now people start to look for new drugs that has multiple proteins that can go for multiple targets. Then answer should be one. Ganoderma Lucidum extract should be one. Right? So it is a natural multiple target. There's no need to look any more further. Okay? I mean, in trying to get scientists paying billions of dollars to crack it. I mean, no doubt the fact that over the years, we always talk about Ganoderma can be supplemental to cancer treatment. No doubt, yes, correct, it should be supplemental to cancer treatment, but maybe it should be also be a primary therapy. Now that we know more about it, right? Of course, I mean, we have to understand that the definition of cancer treatment. When somebody says it's remission and things is growing not at anything less than 12-15% is considered remission. So from that research that we see here, and those graphic representation I showed you just now, right? The tumor size has reduced. Isn't that anti-cancer? Right? Why are we still trying to look for targeted drugs? Immunotherapy, phototherapy, right? So I think network pharmacology is a good way for us to explain to our potential consumer the effects of uh, the pharmacological action mechanism of GLE. Okay, with that, I end my sharing. Thank you. Bye.
Okay, thank you.